So let's get started. 90% of all drug candidates fail. Yeah. Hi, my name is David. I'm the co-founder and CEO of BiolomiX. I'm here representing today myself, as well as my co-founder, who's currently in stealth mode, but this much can be said, is really a talented biologist. Together, we have more than 25 years of experience in academia and industry. So about a year ago, my co-founder and I got to talking, discussing the patented method I developed during my postdoc. We realized that our unique combined skill set in biology, bioinformatics, and data science, together with this method I developed, could be the answer to one of the greatest challenges in the pharma world, and could revolutionize how drugs are developed, going from preclinical, pre-IND data to accurately predict clinical outcome of drug candidates using AI, and flipping this 90% failure rate that I mentioned before into a 90% success rate. And specifically, I'm talking about toxicity and efficacy of drug candidates. So in an ideal world, there exists a linear relationship between mouse and human, so that results obtained from experiments run on mice can be translated into human biology. Thank you.
Wow.
It gets better, it gets better. However, a mouse is not a very little person, and results obtained from mice cannot be translated into human biology. So, go. In reality, what drug or pharma companies do, they start a drug development with some sort of cell culture experiment. If that goes well, then they go over to mouse. And then they throw in a lot of money, and then they go over to a large animal model. That usually doesn't give them the results they were hoping for, so they go back to mouse, and then they iterate between various in vitro and in vivo systems, and then they throw in some more money, and then they go over to another large animal model, and then when they finally get through all these stages, they finally get to human. Right. And at the end of the day, you get a very frustrated pharma industry who has no idea how to deal with this issue. So every year, billions of dollars are spent on drug candidates, of which the majority never reach the market. It takes, on average, 15 to 20 years for the development of a new drug. And despite doing this process now for decades, the failure rate is still over 90%. So we all know that the drug industry is huge, generating a revenue of $1.5 trillion. That's a billion with a T. Sorry. $1.5 trillion per year, of which around 1/4 is still channeled into drug development. I know what you're going to ask, but David, there are already some companies who are trying to aid in the drug development process. That's right. So there are basically two types of companies. One, using data-driven approaches and allowing the computer to analyze and categorize the data. And the other one, using knowledge-based approaches, where the decision-making on how to categorize the data is based on results that have already been published. Our method is more effective and better because we use the best from both worlds. So we're going to, using proprietary technology, we're harnessing the power of network analysis and AI techniques to develop a platform that can bridge the translation gap from clinical data to clinical outcome. The platform is capable of ingesting data of all modalities. It's composed of various components, but at the heart of our platform sits our MVP. I always say minimal viable product, but also our most valuable player, the activity score. And this is what differentiates us from others. And sometimes it's easier to explain what something does by showing what it doesn't do. Okay, so the common approach to determine the toxicity and efficacy of a drug candidate is to take some quantitative measurement of biological feature and then assess the differences using statistical analysis. So in this case, up here, we would say there's no significant difference between the treatment and between the control and the treatment group, and therefore the drug doesn't have an effect. In this example down here, we see there's a significant increase between the control and the treatment group, and therefore we'd say the drug has an effect. It's basically agreed upon in the scientific community that statistics is not enough to really understand and explain biology. However, it's still considered the gold standard for biological experiments, and most certainly to determine the toxicity and efficacy of drug candidates. We at BiolomiX say it's time to rethink how biology is measured. It's time for a paradigm shift. And instead of using quantitative measures, let's use the activity. And that's what we call the activity score. So in this case, although quantitatively not significantly different, the activity score would say that the drug is inactive in the control group, but very active in the treatment group. Or in that case down here, we would say that the drug is in the control group, it's very active, but in the treatment group, it's very inactive, although the opposite trends are visible using quantitative measures. The activity score brings us one step closer to causality, and that's why it can identify hidden liabilities of drugs. So let's go into a real-world example. In 1985, Takeda Pharmaceuticals patented a pioglitazone, a drug to treat type 2 diabetes. They went through all the preclinical trials and then the clinical trials and finally got the FDA approval, and then the drug was released into the market. Pretty soon after that, patients started to suffer from toxic side effects that were not discovered before, and in some cases led to heart failure, and which led to death of patients. Pretty soon after, thousands of lawsuits kept pouring in, and the drug was suspended in many countries in the world, and that was a huge disaster for Takeda, and they just settled in 2014 for a sum of $2.7 billion to settle the case. So we used a study from 2018 where obese rats were treated with pioglitazone, the same drug, and metabolic profiles were taken from metabolic profiles were taken from liver samples and from serum. And we took these samples and fed them into our MVP, into our activity score. And we realized the drug does what it's supposed to do. It activates and deactivates insulin and sugar-associated pathways. However, we also realized opposite trends of the prostanoid biosynthesis pathway in the liver and the serum, which is known to cause heart failure. So we tried to replicate these results with common methods, all of which failed to do so. So had the pharma company used our method instead, they would have discovered those toxic side effects already during preclinical trials and could have spared the lives of thousands and saved themselves a big, big headache. So our platform is going to give a huge advantage to the pharma industry. Obviously, it's going to reduce costs and increase profit margins by that, but at the same time, it will also ensure higher reliability of the drugs that reach the market. But not only for the pharma industry will it be beneficial, also for the patients. It will ensure better treatments, reduce adverse effects, and at the end of the day, save lives. I hope this is a method you agree with. We've surrounded ourselves with a great team of advisors, with some of the leading scientists in artificial intelligence in the world, but also advisors who are very, very experienced in business development and finance. We're looking for a pre-seed of $1.2 million for 1 year. And this is mainly to build a team so my co-founder can finally come out of stealth mode, and we also will identify a chief developer, and then we'll develop an SaaS API-based version of our MVP. We already have a raw version of it, and as you can see, we already have some traction with the pharma industry. We have 3 verbal endorsements from senior executives from 3 of the biggest pharma companies in the world, and they agreed that they would supply some data so we can run them through our pipeline, and this data is basically drugs that pass the preclinical stage but failed at the clinical stage, and they want to see how we can solve this. And at the end, we will close with the seed round. If you have any more questions about BiolomiX and what we're trying to develop and here, please talk to me. Happy to. Thank you very much.